Assalamualaikum and very good morning to all of you. We from Group 5. So today we are going to present the invertebrate evolution. So this is our group members. In this chapter, we are going to cover the evolution of animals and the simplest invertebrates. Okay. When you finish the evolution of animals, you should be able to list 3 common characteristics of animals that are not found in other multicellular eukaryotes. You are able to summarize the colonial flagellate hypothesis as it relates to the origin of animals. You can distinguish among the different body plan of animals, and you can compare protostome and deuterostome development.
Hi, my name is Muhammad Akhil Kudni from Group 5. Today, I am going to present the evolution of animal. Okay, in domain Eukarya, there are 3 kingdoms, which is kingdom Animalia, kingdom Fungi, and kingdom Plants. Okay, we human are classified under the kingdom Animalia. For fungi, there is mushroom, and for kingdom plants, there is angiosperm, the plants which bear flower. Okay. We are going to discuss the characteristic of animals. Okay. First, animal cells lack a cell wall. Second, animals are consumer, while fungi are saprotroph. Actually, both animals and fungi are both heterotroph, but animals are a little bit different as animal ingest food and digest it internally, while fungi they absorb food externally and absorb nutrients. Uh, the third one is animals are mobile, have nerves and muscles, and reproduce sexually. Okay, let's look first for the muscle and nerve tissues. Muscles and nerve tissues allows motility and flexible movements. Motility and flexible movements enable animals to search actively for food and to prey on other organisms. Flexible movement also enable animals to seek mates, shelter, and suitable climates behavior. Okay, let's see for the here we have bear. Okay, bear ni beruang ni merupakan consumer ataupun kita boleh kata holozoid sebab dia obtain nutrition from external sources. Dia makan mikroorganisma lain. So, dia ni kategori dia adalah holozoid consumers. Okay. Disebabkan ada nerves, muscle and nerve tissue ni hadir, bear ni dapat mencari makanan, dapat bergerak untuk mencari makanan dan untuk meneruskan hidup. Okay, sini kita tengok swan. Nerves and muscle tissues ni enable swans to seek mates. Dan kat sini kita ada cendawan. Ni merupakan cendawan busut yang selalu kita jumpa lepas hujan, dalam sehari 2 hari lepas hujan, ataupun nama saintifik dia Termitomyces species. Okey, cendawan ni merupakan saprotroph sebab dia serap nutrien dekat tempat-tempat lembap. Contoh macam kayu lembap. Okey. Okay, then animals have a variety of life cycles. Okay, most of animals reproduce sexually. Some reproduce asexually and some combine both life cycle. Okay, many animals have a diploid life cycle, but some animals also have other life cycles such as haploid or haplodiploid. Okay, animals undergo a series of developmental stages to produce an organism that has specialized tissue. Okay, so here to end zygote. Actually to end zygote ni in most of animal, because most of the animal reproduce sexually, and this zygote to end zygote will undergo development to produce a multicellular organism that has specialized tissue. Okay. Then animals are descended from a single common ancestor, thus forming a single lineage on the tree of life. Single common ancestor ni refer to choanoflagellates. Dan choanoflagellates ni dia akan membentuk cabang-cabang dia lah yang membentuk lineage ni kat tree of life. Okay. There are two main branches in animal lineage, which is vertebrates and invertebrates. Okay, vertebrates ni are animals that have spinal cord. Haiwan yang ada tulang belakang, backbone. Manakala invertebrates, dia ada tulang belakang. Okay. So, kat sini kita nampak ada cheetah kat sini. Oh, sorry. Ada cheetah kat sini. Cheetah ni bukan vertebrate sebab ada tulang belakang. Tapi Kurita ni, octopus, is invertebrates because it does not have backbone. And here we have sea anemone. Actually, sea anemone is special because it reproduce both asexual and sexually. Sea anemones reproduce depends on where it live, depend on the depth in which it live in the ocean. Lagi dalam tempat dia tinggal dekat dalam laut tu, lagi cenderung dia kepada sexual reproduction. Manakala kalau lagi cetek tempat dia tinggal, lagi cenderung dia kepada asexual reproduction. Okay, kemudian kita tengok ancestry of animals. Okay, animals evolved from a protist, most likely a motile protozoan. Okay, colonial flagellate hypothesis states that animals are descended from an ancestor that resemble a hollow spherical colony of flagellated cell. So dia ancestor ni, chronoflagellates ni, dia akan membentuk satu sphere bulatan yang ada macam hole, macam lubang kat tengah. Okay. Choanoflagellates most likely resemble the last single cell ancestor. Okay, the molecular data tells us the closest living relatives of animals. Okay, the stated choanoflagellate is a single cell. It has a diameter around 3 to 10 micrometer and have a flagellum surrounded by a collar of 30 to 40 microvilli. Okay, the movement of the flagellum creates water current which pull the protist along. Then the microvilli around the flagellum will engulf bacteria and debris from the water. Okay, choanoflagellates can also exist as colony and can be found together at the end of stalk or simply clump together like a bunch of grapes. Okay, kita tengok rajah ni. Yang ni adalah satu sel choanoflagellates. Okay, dia ada nukleus dan ni body cell dia. Okay, yang ekor ni kita panggil flagellum. Bila dia bergerak je, dia akan menyebabkan water current lah. Kemudian kat sini, kat collar ni, terletak microvilli dalam 30 ke 40 microvilli. Okay. Dan kita tengok colonial flagellate hypothesis. Okay, dia start pada satu je, unicellular choanoflagellate. Kemudian dia akan panggil kawan-kawan dia pada family dia untuk berbentuk satu aggregate atau group kumpulan. Dan kumpulan ni dia akan membentuk satu holosphere lah. Unspecialized vegetated cells ni dia akan form holosphere yang mana tengah ni kosong. Dia macam lubang, hole kat situ. Dan sebahagian daripada chronoflagellates ni dia akan form specialized reproductive cells yang warna purple ni. Lepas dia form specialized reproductive cell ni, dia akan mula untuk melipat. Sel ni akan melipat. The cells begin to fold in. Lipatan sel ni akan menghasilkan satu specialized tissue. which this specialized tissue has its own particular function. Okay. The colonial flagellate hypothesis is also attractive because of its implication regarding animal symmetry. This animal symmetry, we're going to discuss it later. Okay. Then we go with the evolution of body plants. Cambrian period present all the various body plant around 500 million years ago. Okay, as animal develops, there are many possible outcomes regarding the number, position, size, and pattern of its body parts. Means, semua ni possible outcome ni mungkin dari segi nombor, posisi, size dan pattern dekat bahagian badan dia mungkin berubah. Sebab apa berubah? Disebabkan slight shift of DNA code and expression of hot genes. Akan menyebabkan perubahan masa development tu. Okey, contoh dia kita tengok kat sini. Sini kita ada macam multi-limb crustacean-like ancestor. Kita ada serangga, maksudnya nenek moyang serangga tu dalam 400 juta tahun yang lalu lah. Serangga ni, dia mengalami mutasi dekat UBX hot genes dia. UBX ni untuk genes dekat serangga. Okay. Dia mengalami mutasi dan menyebabkan limbs in abdomen dia untuk repress. Jadi ingat kaki dia panjang-panjang ni, yang banyak ni, tiba-tiba jadi mutasi dan development tu, outcome development tu, dia merubah kaki dia jadi 6. Ingat kaki dia pun berubah, ada sendi. Okay. That's all for my parts. I will pass the presentation to my teammates. Thank you.
Thanks to Yad. Hi, I'm Muhammad Madi bin Azam Zaki, and I'll continue the presentation. Now let's move on to the phylogenetic tree of animals. So, the fossil record of the early evolution animals is very past. Then the hypothesis of the evolution is based on the combination of morphological characters of living and fossil animals, developmental homologies, and molecular DNA sequence. The concept is, the more similar their DNA sequence are, the more closely related they are. And as a result, updated phylogenetic tree are quite different from the theses that are historically based.
Bacteria and archaea are the unicellular prokaryotes we talked about, and eukarya is all eukaryotes. This means unicellular protists as well as all multicellular life on Earth, including every plant and animal there is. Within the domain eukarya, there are several kingdoms. Animalia contains all animals. Plantae contains all plants. Fungi contains all mushrooms and molds. And Protista contains all the rest, although many biologists don't recognize this as a valid kingdom any longer. Within each kingdom, there are several phyla. Within the animal kingdom, there are 9 major ones. There is the phylum Arthropoda, which contains all kinds of bugs and lobsters and creepy crawlies. Mollusca, which contains octopi and snails and other such things. And most notably, there is Chordata. This phylum includes any animal with a spinal cord, and the divergence of vertebrates and invertebrates was a major step in evolution. Within each phylum, we can find different classes of organism. Chordata contains the class Reptilia, which are reptiles like crocs and snakes and lizards. Amphibia, which are amphibians like toads and frogs. And Mammalia, which are all the mammals, from kangaroos to elephants. Humans belong to Mammalia, and within this class there are a number of orders. The order Cetacea contains all the whales and dolphins. Carnivora contains things like bears and weasels. Then there are the primates. That's got everything from orangutans to lemurs, and humans are in there too. Within the order of primates, there are several families. The family Hominidae contains all the great apes, so that's us again. Within Hominidae, every organism belongs to a particular genus, and that's where we diverge from the apes. Our genus is called Homo, which means man in Latin. Within a genus, there are several species, and all the species within the genus Homo are now extinct, except for Homo sapiens, and that's us. The species that came before us, Homo erectus, Homo habilis, and many others, are all gone. And Homo sapiens, which means wise man, sits at the very top of the tree, the only species to have evolved an intelligence that spawned complex civilizations and information storage, first as written language, and eventually the omnipresent internet.
Talk about the type of symmetry for animal development. But first, we must understand what this symmetry means. Symmetry comes from the Greek word, which is first, syn, which means together, and metron, which means measure. And basically, symmetry means a pattern of similarity that is observed in object. And for this topic, of course, for the animals. And there are three types of symmetry for animal development. First, we got symmetry, which obviously means lack of symmetry. The animal have no particular pattern of body shape. For example, for sponge, if we give it a cut through from any direction, we're not going to get a same half. And then, second, we got radial symmetry. For this animal, their bodies are organized circularly, similar to a wheel, and a longitudinal cut through the center point produce two identical halves, example, snadrons and xenophores. So if you look at the diagram, for example, for this cut through, if you give it a cut through like this, then we're going to have the same side from this side and this side. And the same will happen for each cut through. And this we got radial symmetry. Third, we got bilateral symmetry. And the animals have definite left and right halves, and only a single longitudinal cut through the central point produces two identical halves. So for dorsal and ventral cut through, we're gonna get the same side of the turtle, which consists of the right side and the left side. Then, the animal have defined anterior and posterior ends, and forward movement is guided with the anterior end. So anterior is the front of the animal, and posterior is the back of the animal. And not to forget, they are accompanied by civilization. What is civilization? Civilization means localization of a brain and specialized sensory organs at the end of the animal. So for this slide, we're going to talk about embryonic development. So embryological development is where true tissues develop in the more complex animals. And the first appeared layer called germ layers. So what is germ layers? Germ layers consists of all of these layers, and it gives rise to the organs. First, we got embryo with two tissue layers, which we call as diploblastic, and this layer consists of ectoderm and endoderm layers. So we can observe the diagram. For diploblastic, we got ectoderm in the outer layer, and in the inner layer we got endoderm, and between them we got non-living layer. Second, for embryo with three tissue layers, we call them as diploblastic, and it consists of ectoderm, mesoderm, and endoderm. So look at the diagram for diploblastic. In the outer layer we got ectoderm, and in one we got endoderm, and between them we got mesoderm. So there's a difference between diploblastic and diploblastic. And notice that all animals except sponge, xenophores, snadron, and protozoans are diploblastic. And the diploblastic animals are either protozoans or deuterozoans. And what is protozoans and diploblastic? So protozoans come from the Greek word proto, which means first, and the word stoma, which means mouth. But for deuterozoans, it comes from the words deuter, which means second, and then the word stoma, which is mouth. And for protozoans, the animals that have the mouth that develops prior to the anus. And opposite to the protozoan, the deuterozoans mean the animals that the anus that develop prior to the mouth. And to differentiate both of these protozoans and deuterozoans, we're going to have a look at the next slides. So in next slides, we're going to talk about the three major events that will differentiate between both of the protozoa and deuterostomes. And the three major events consists of cleavage, blastula formation, and column. So for cleavage, what is cleavage? Cleavage is the first development event after fertilization cell division without cell growth. For protozoans, spiral cleavage occurs, and the fate of the cell is fixed and determinate, and each cell can develop in one particular way. But for deuterostomes, radial cleavage occurs, and the fate of the cell is indeterminate, and each cell can go on to become a complete organism. Moving on, we're going to talk about blastula formation. Yet, blastula or a hollow sphere of cells produce an opening called the blastospore as development proceeds. And in protozoans, the mouth appears at or near the blastospore. But for deuterostomes, the anus appears at or near the blastospore, and only later does a second opening from the mouth. And next, for column development, for certain protozoa and all deuterostomes have a body cavity lined by mesoderms called a column, which comes from the Greek word kolyma, which means cavity. The column in this group is a true column, and for protostomes, the mesoderms arise from cells located near the embryonic blastospores, and splitting occurs that produce the column. But for deuterostomes, the column arises as a pair of mesodermal pouch from the wall of the primitive gut, and the pouch enlarges until they meet and fuses. So, three of these major events will differentiate them.
Started dividing and cleaving through mitosis, resulting in 2, 4, 8, 16 cells, until it creates a solid ball of 32 cells. This is actually a morula, or morula, at least according to this guy.
Morula, or morula.
And the morula actually looks a lot like a raspberry or a mulberry, which is what it's named after in Latin. Juicy. Morula pie. Oh God. They're gonna ban us from schools. As more cells are created, the solid wad of cells begins to secrete a fluid that forms a space in the center, resulting in a hollow sphere of cells called a blastula. Okay, so pay attention because here's where we're gonna get down to the real business. Most animals that you just sort of think of off the top of your head have a mouth, right? And by the same token, most of them have an anus. And yeah, you go ahead and get your giggles out now because I'm going to be saying anus a lot in this video. For example, right now, anus. So most animals have a mouth and an anus. Wait for it, unless you're a sea sponge. Sponges don't have a mouth or an anus. And there are also other animals like your sea anemones, your jellyfish, your corals that have just one hole that serves as both mouth and anus. The process of forming this tract is called gastrulation, and it's kind of a big deal. So when we left our little blastula, it was still just hanging out, a little round hollow ball of cells. Gastrulation begins when an indentation starts to form at a single point on the blastula. This place on the blastula that starts to invaginate or fold in on itself is called the blastopore. Now for animals whose mouth and anus are the same thing, this is where the development stops, which is why they only have one hole for all their business. But in everything else, the invagination continues until the indentation makes its way all the way through and opens on the other side, creating what is essentially a hollow bead made of cells. Now we have a gastrula. Now two different things can happen at this point depending on what kind of animal this is going to be. It can either be an animal whose mouth is the orifice that's formed by the blastopore, called a protostome, or one whose anus is the structure that's created by the blastopore, and that's called a deuterostome. So guess which one you are. You just, write it down. I want to see your guesses. Chordates, that is to say all vertebrates and a couple of our relatives like starfish, are deuterostomes, meaning that we were once just a butthole attached to a little wad of cells. And that includes you and me. Congratulations! And hopefully you're getting the idea here. The formation of the digestive tract is the first thing that happens in the development of an animal. And it happens to every living thing, whether it's going to be a tardigrade, or a polar bear, or a T-pain. The miracle of life! Now, so far, the little hollow bead of cells is basically 2 layers of tissue thick, an outer layer called the ectoderm, and an inner layer called the endoderm, and these are called your germ layers. For those organisms that stop developing at this point with that classy mouth-anus combo, they only get 2 germ layers. They're called diploblastic, and they were born that way. It's totally okay. But for us more complex animals whose mouths are separate from our anuses, we develop a 3rd layer of tissue, making us triploblasts. Here, the ectoderm is going to end up being the animal's skin and nerves and spinal cord and most of its brain, while the endoderm ends up forming the digestive tract, the esophagus and stomach and colon and stuff. And in addition, some of the cells start breaking off between the endoderm and the ectoderm and form another layer called the mesoderm. These cells will eventually end up as the muscles and the circulatory system and the reproductive systems, and in the case of vertebrates, most of the bones. So what's our embryo looking like now? Awesome.
So that's the end of the video. Now I will pass the presentation to Nazra.
Hi, I am Siti Nazra and I will continue the invertebrate evolution with the simplest invertebrates. So, from the learning outcomes, you must be able to explain why the sponges are considered to the simplest animals and how they reproduce, feed and raise fires. And lastly, you must be able to compare the features of ctenophores to those of cnidarians, such as hydras.
Okay, this is the sponges, which known as the pore-bearing. The sponges is under the kingdom Animalia with phylum Porifera. So they are multicellular which lack of tissues and organ and have the asymmetrical shape, and the lack of tissues and organs means the lack of nerve system and don't have a muscle cells. So which mean they do not move and stay still at the bottom of the ocean. So we can see the sac-like body is perforated by many pores, and they are largely marine animals which great in size and shape. So they have a canal system of pores which allows water to move through their body, and they are filter or known as suspension filter, which will filtering the food particles, and like example, the microscopic food particles that pass between the microvilli are engulfed by the collar cells that I will be explained more, and the collar cell will digested by them in food vacuoles. So the canal system of pores. So we can see the interior of the canals is lined with the flagellated cells that resembles the choanoflagellates. So the choanoflagellates help the sponges to have a cellular respiration. So this choanocyte will act as pumps to bring water, and the food will be filtered for the sponge get the nutrients. So the choanocyte will create water currents for filter feeding, which flow through the pores into the central cavity and out through the osculum, the upper opening of body. The sponges have a specialized cells, which the amoebocytes here, and they will transport nutrients from cell to cell, and the choanocyte will create current for filter feeding by acting as a pump. So the sponges have a skeleton, has an endoskeleton type. The endoskeleton type will prevent the body from collapsing because inside the sponges they have the spicule, which is only small and have a needle shape which is bony class, and they are sponging sponges. And the spicule will help the sponges to discouraging the predators because the spicule will produce a toxics. So next is the reproduction.
Okay, the reproduction of sponges. The sponges had an asexual and sexual reproduction, which the asexual, they had a fragmentation and budding process. So the fragmentation, the body of the sponges, like the parents, will break off into pieces. So next, they will regenerate a new sponges. So the budding means a small protuberance appears and gradually increases until a complete organism forms. So they will produce colonies of sponges. The next simplest invertebrate is ctenophores, which are known as comb jellies under the phylum Ctenophora. So the comb jellies are solitary and mostly free-swimming marine invertebrates. The comb jelly, usually found in warm waters, means they can easily move around the environment, like sponges, which they just still at the bottom of the ocean. So the ctenophores represent the largest of the simplest invertebrates, and they move around by beating their cilia. The ctenophores, also known as bioluminescent, because body is made of transparent, which are known as a mesoglia. The mesoglia is the jelly-like substance, and ctenophore also have a coloblast, which is the sticky adhesive cells that help them to capturing prey, and they don't need sting cells like others. Okay, the last simplest invertebrate is cnidarians. Cnidarians is known as the stinging nettle because they have a cnidocytes, which the cnidocyte has a fluid-filled capsule called a nematocyst. So the cnidarians have a radial symmetry and have a tubular or bell-shaped animals. So they're usually found in shallow coastal waters or some in freshwater, brackish, and oceanic forms. So the specialized cnidarian cells, which is the cnidocytes that I have told before, and the nematocytes. So the outside is the cnidocyte and the inside is the nematocytes. So the stinging structure inside contains a long, spirally coiled hollow thread. So when the trigger of the cnidocyte is touched, the nematocyst is discharged. So some threads merely trap a prey and others have spines that penetrate and inject paralyzing toxin. So we can see the body of cnidarian have two layers of sac. So the outer tissue layer is act as a protective epidermis that derives from ectoderm, and the inner tissue layer is derived from endoderm, which will secrete a digestive juice into the internal cavity called the gastrovascular cavity. So the gastrovascular cavity will help in digestion of food and circulation of nutrients. And they also serve as the protective hydrostatic skeleton because of their fluid field. And it will offer some resistance to the contraction of muscle but permits flexibility. And we can see the mesoglia are separating the both tissue layer. So the cnidarian basic body forms. So they have polyp form and medusal form. So we can see the polyp is tube-shaped body and upward mouth, and they reproduce asexually. Meanwhile, the medusa form is umbrella-shaped body and downward mouth, and they have more mesoglia than a polyp. So their tentacles are concentrated on the margin of the bell, and they reproduce sexually. So at one time, both body forms may have been a part of the life cycle of all cnidarians. When both are present, the animal is dimorphic because of the sessile polyp stage will produce medusae by asexual budding, and the motile medusan stage will produce eggs and sperm and will producing the new cell of cnidarian.
So in some cnidaria, one stage is dominant and the other is reduced. In other species, one form is absent altogether.
Hi guys, my name is Nurul Nabila binti Mahza Sharil, and I am here today to explain the last part of the topic, which is cnidarian diversity. The first cnidarian is sea anemones. Sea anemones is a sessile polyp that live attached to submerged rocks, timbers, and other substrates. Most sea anemones range in size 0.5 to 20 cm in length and 0.5 to 10 cm in diameter and are often colorful. For example, these three pictures show that it has a lot of colors. Next, their upward turned oral disc contain the mouth and is surrounded by a large number of hollow tentacles containing nematocysts. This means in the middle of the sea anemones, it contains the mouth and is surrounded by a large number of hollow tentacles. As you guys can tell from the picture, it has a large number of hollow tentacles that contain nematocysts inside it. Next, corals. Corals resemble sea anemones encased in a calcium carbonate limestone house. This is the limestone house. The coral polyp can extend into the water to feed on microorganisms and also retreat into the house bag for safety. Coral reefs are built from the slow accumulation of limestone produced by corals. Next, some corals are solitary, but the vast majority live in colonies that vary in shape from rounded to branching. These are the examples of the branching corals. As you guys can tell, it has a lot of different shapes to it. Next, these are the examples of solitary corals. In the middle of the solitary corals contain the mouth of the polyp. Next, corals are the animals that build coral reefs. They exhibit elaborate geometric design and stunning colors. For example, on this picture shown here, there are a lot of geometric design and stunning colors. Coral reef ecosystems are very productive and a diverse group of marine life calls the reef home. This picture shows the reef home because there are a lot of group of marine lives. Next, jellyfish. In true jellyfish, also known as sea jellies. The medusa is the primary stage and polyp remains small. Jellyfish are zooplankton, depends on tide and currents for their primary means of movement. This sentence means that jellyfish moves around depends on their tides and current. They also feed on variety of invertebrates and fishes and are themselves are food for the marine animals. Next, hydrozoans. Hydrozoans have a dominant polyp stage. Hydra, which is this one, Portuguese man of war, which is this one, are hydrozoans. It is a colony of polyps. The original polyps becomes a gas-filled float that provide buoyancy, keeping the colony afloat. So this is the gas-filled polyp. It helps to keep the colony afloat on the water. Other polyps which bud from this one are specialized for feeding and also for reproduction. A long single-tentacled arm with numerous nematocysts arise from the base of each feeding polyp. So these are the tentacles. So it has a lot of numerous nematocysts arise from the base of each feeding polyp. So swimmers who accidentally comes upon a Portuguese man of war can receive painful, even serious injuries from the stinging tentacles. So I include the examples of a man who sting by the Portuguese men of war. Next, a typical cnidarian, which is hydra. Hydras are likely to be found attached to underwater plants or rocks in most lakes and ponds. So this picture here shows that it is attached to underwater plants. The body of a hydra is small tubular polyp about 1/4 of an inch in length, which is very, very small. The only opening is in raised area surrounded by 4 to 6 tentacles that contain a large number of nematocysts. For example, in the picture number 1, it has 6 tentacles. On the picture number 2, have 4 tentacles. And in the tentacles, they have contained a lot of number of nematocysts. Next, the microscopic anatomy of hydra species. The cells of epidermis are termed epitheliomuscular cells. Under epitheliomuscular cells, it contains muscle fibers, nematocysts that contain cnidocytes, which is this one, sensory cells that make contact with nerve cells within a nerve net. So these are the example of the hydra species nervous system. So these interconnect nerve cells allow the transmission of impulse in several directions at once. In the nerve net, there are a lot of nerve cells. So the interconnected nerve cells allow the transmission of impulse in several directions at once. The body of hydra species can contract, also can extend. The tentacles that ring the mouth can reach out and grasp prey and discharge nematocysts. So these are the tentacles, and this is the mouth of hydra species. So it can reach out and grasp prey and discharge nematocysts from the tentacles. Next, hydra life cycle. So hydra life cycle has two, which is reproduce sexually and asexually. So we go for sexually. Sperm from a testis swim to an egg within an ovary. So this is the sperm will swim to an egg within an ovary. The embryo encased with a hard protective shell that allows it to survive until conditions are optimum for it to emerge and develop into a new polyp. So on female hydra, it will encase within a hard protective shells that allow it to survive until it is optimum to develop into new polyp. Next is reproduce asexually. It is produced by forming buds here. And it will develop into a complete animal and then detach. Interstitial cells of the epidermis are capable of becoming other types of cells. For example, an ovary, or it could be a testis. Next, I will conclude the comparison between the sponges, ctenophores, and cnidarians. So body plans for sponges are asymmetry. Body plans for ctenophores are biradial symmetry. So biradial, what is biradial symmetry? Biradial symmetry is a radial plus bilateral symmetry. It is a combination between two symmetry. For cnidarians, it is a radial symmetry. Sponges feed feed using filter feeders, and the digestion takes place in amoeboid cells. Ctenophores feeding, they capture their prey using coloblasts. What is coloblast? Coloblast is a sticky adhesive cells. And the digestion takes place in gastrovascular cavity. For cnidarians, they capture prey with nematocysts and tentacles, and the digestion takes place in gastrovascular cavity, which is same as ctenophore. Movement. How does sponges move? So it moves by sessile. What is sessile? Sessile is stay or attached to one place. Ctenophore move by swim by comb plates. Cnidarians move by sessile or free floating. Response to stimuli. How sponge responds to stimuli? Sponge doesn't have any nervous system, so cells react to stimuli by closing their pores. For ctenophore, they have nervous system, so it produces light, which is called bioluminescent. For cnidarians, it is simple nervous system consists of nerve net. For reproduction, sponges have two reproduction, which is asexual fragmentation and budding, and sexual reproduction. For ctenophores, they don't have any asexual reproduction. It only has sexual by fertilization. Cnidarians reproduction, Medusa reproduce sexually. For polyp, reproduce asexually by budding. That's all from our group. Thank you.